team Predicting Microbial Growth from Data Plus 2025, and today we're going to be introducing to you MicroNet. MicroNet is a web-based app used to assist researchers in predicting microbial growth through the Gaussian process model. It's important and helpful and useful because it allows researchers to do low-code Gaussian process modeling, which is more flexible and also allows the model to handle better conditions under stress. And to preface this, all of this is being done locally through Docker, but once the lifecycle of the app is completed, it should be hosted online. So we're going to begin by getting into the app by putting in our email.
Now let's look at the different file types that MicroNet support. First, you need your data files. These will be generated through the researcher's plate readers. And these just include the measurement of the concentration of the microbes over time. Second, you use a mapping file to label your different data files. It specifies what each individual wells contains. And with our recent improvements, you can actually match your different data files to the corresponding mapping file you're interested in. And you can do multiple files together. Next, in addition to mapping file, you can also label with a meta file. So a meta file actually labels the entire plate rather than labeling individual wells like the mapping file. So you can see here, it will label everything in plate 1 as this and label everything in plate 2 as this. Next, if you want to adjust how often your bacterial concentration is measured, rather than the default 600 second, you can upload a time interval file. And let's upload our data and move on to the analysis.
So for analysis, we're given a choice of analysis to select. The first one being raw data plot wells, which will plot a growth curve for each well within your data, giving us a preliminary analysis.
Second, we have the most robust and advanced Gaussian process plot. Based on the condition you pull by, it will generate a growth curve based on Gaussian process regression. Here we choose isolate. You can also further subset the data to reduce the amount of data you're interested in.
Next, we have fit statistics, which produces a summary table of the model's growth curves based on 5 metrics, being area under the curve, growth rate, carrying capacity, lag time, and doubling time. We can also choose to pool replicates, which will combine these metrics and create a unique combination based on those variables to reduce the number of rows, basically creating a more concise fit statistic summary.
Next, we have heat mapping. And here we can choose the specific values we're interested in and determine our different axes. Now here let's have substrate as our x-axis and have isolate as our y-axis. Next, we have test hypothesis, which we'll select 2 specific conditions we're interested in and compare the Gaussian process regression model of the 2. Let's choose 2 different isolates here, CD1007 and CD2001. Next, we have compare parameters and new features added. It is very similar to the previous test hypothesis options. However, rather than comparing and determining if there's functional differences between the Gaussian process regression modeling of the 2 curves, it will actually first generate different summary statistics we mentioned before and compare if there's significant differences between the different summary statistics. Let's compare using substrate with glucose and fructose. Now let's review all our different choices. Here are all our files. Let's write 2 data files, 2 mapping files, different analysis, and let's start our job.
So when we start our job, we will see that we'll end up getting a confirmation email sent to our email to show that the job has done successfully, and then we must wait for the job to process.
Here, since we already generated these files, because it takes about 15 to 20 minutes to get all the files ready for you, so we just kind of download these previously. Here you can see we have the 2 raw data plots since we have 2 different plates. Next, we have the Gaussian process regression plot since we pull by isolate. So for each individual isolate, it will generate a Gaussian process regression based on the Gaussian process output. And it will also model the different variants of the individual curves. Next.
We also have the heat map, which has the substrate and the isolate basically plotting the strength of the metric of those variables within each plot of the heat map based on growth rate.
Next, we have fit summary, which is based on fit statistic options. Since we didn't pull by anything, it will actually generate the 5 different summary statistics for every single well in the 2 plates. Next, we have our test hypothesis, which will have 2 options and outputs actually. So here we compare the 2 isolates based on their OD and curve. Next, we'll actually get the summary between the 2 to see if there's significant differences between the 2 curves. We have a log Bayesian factor, which basically tells us there's a lot of difference between CD 1007 and CD 2001. Lastly, we have our compare parameters output, where we compare the glucose and fructose. And you can see for each individual summary statistics, it will help us kind of determine whether if there's significant differences between the area under the curve of glucose and fructose and etc. And this will be really useful for the researchers to make different conclusions whether the specific conditions or genes they're interested in is actually affecting how the microbes grow. And this is our work for the summer of 2025. Thank you very much.